Hi, everyone. Thanks for having me here today. So yeah, I'm Alexis. I'm Director of AI at Nostrum. Yep. Should be. Yeah, there we go. So we are a spin-off of two research centers here in Barcelona, the Barcelona Supercomputing Center and the Institute of Biomedical Research. And basically, we were born 7 years ago, and with a clear objective. So to create technologies that could help biotech and pharma achieve their goals in specific scientific projects. On this journey of developing technology, we have been investing a huge amount of our revenue, so 35%, on new technologies, new R&D. And this is what brings me here today. As a director of AI, most of this investment that Nostrum did helped see the born of many of the technologies that I'm going to show today. So what's our main goal here, our main purpose? So it's the following. Does it display? No. So I'll walk you through that. So basically, this was supposed to be a video of a protein. Okay, so this guy in here, this loopy guy in here, it's a protein. This guy, it's not static, it's dynamic. Every time that it changes conformations, we can see new binding spots where drugs, molecules can get into and inhibit this protein. Usually, when we get sick, it's because one of these loopy guys has a mutation and it makes the wrong function. So our goal here is to design and to find these guys, this not so loopy guy, to bind into one of the regions of this protein to inhibit that, right? And this is mainly what all of our technology is about. But the way that we approach technology, and especially AI, changed over the years. So especially with two main realizations. So one is the advent of combinatorial chemistry. This is the possibility of having molecules that are just synthesized and they're waiting for you in a shelf somewhere, and that you can buy and test, and you can also try virtually with computers. So in 2010, we had 16 million available. In 2017, we had 10 billion available. In 2022, 50 billion. And now, 3.4 trillion molecules that we can work with. This is a huge wealth of possibilities. It also happened the same with proteins. So the protein data bank, which is the major database for storing these crystallographic experiments, so 3D resolutions of our proteins, the proteins in our body and other living organisms, it has grown a lot. It's been growing a lot on the last years. Now we are on the verge of 230,000 different structures that we can use to interrogate with our molecules, with our trillions of molecules. It's 230K for the number of proteins that have been resolved experimentally. But again, AI has helped us to grow this number to the 200 million. So imagine the combinatorial power of 200 million different proteins and 3.4 trillion different molecules for treating new diseases, right? But now we're sitting on this wealth of data, but the truth is that it has not been translated yet to drug discovery. So as of now, in the small molecule space, I'm not counting here antibodies, RNA, gene therapies, we have now 650 binding pockets, which are these regions that we need to find in the proteins to inhibit, because we need a molecule to bind there. And they are targeted only by 1,500 FDA-approved drugs, molecules. This is a tiny, tiny space compared to the whole, the vast chemical space that we are able to explore now. So this is where our technologies focus now, to cover this space. So we have been predicting with our technologies 60,000 different pockets, binding regions in the proteome that are targeted now by 0 molecules. Our goal here is to expand the number of small molecules that can target these 60,000 different pockets to attack new diseases, new indications, and also to revisit indications that are already solved, but we can probably solve them better than they are solved now. So our goal is to create an atlas of human drugability, is to pair billions of different small molecules that are, as I said, are sitting on shelves that we can readily buy with 60K binding pockets that we have been able to discover with our technologies. Also, we're going to pair them with the technologies that I will show you now, and that in matters of just days of compute, we can make assessments of how these guys pair with the different proteins, and then get them to the lab to see how they behave in real conditions. So, our powerhouse are protein language models. Just as language models that they learn from English, Catalan, Spanish, we're able also to learn from proteins and create rich and meaningful representations of these guys, of these proteins. Now, this is our core, but then learning from the representations that these protein language models are able to give us, we're able to design models, AI models, that are able to find these binding regions on proteins, and also to create models that are able to assess the compatibility of billions of chemistries for each and every one of these pockets that we're able to find. Also, the other pillar, as I told you, the proteins are not static objects. They move around, they change conformations, and with this movement, new pockets come and go. So we're also able to simulate this movement and to create training data for our models. That basically, what we're trying to do here is to create data with simulations, and the more simulations that we're able to have for the proteins, the more conformations that we were able to predict for these proteins. Predictions are way cheaper than simulations, and the more that we're able to train with, the less cost we have for getting these conformations. So we want new conformations to discover new pockets and then assess billions of chemistries in a matter of just days. So what's the catch for us? Because everyone is doing protein language models. So basically, protein language models and language models in general, they assume a minimal unit of information that they need to process and make operations with. For instance, in natural language processing, if I have the sentence, the cat sat, I will just assume that the word is the minimum amount of information. And for protein language models in general, the minimum amount of information is the amino acid, which, fine, but is it really the best solution? So this is what we revisited, right? So we created a new technology that is able to learn on the go while we train these models, which is the minimum amount of information that these models need to train with, right? So it may be just a single amino acid. In this case, it's a nucleotide. It can be pairs. It can be triplets. It can depend on the sequence. It can depend on the protein family, right? So we're able to learn that on the go. Now, we can couple that with normal architectures, normal transformer, normal protein language model architectures, and we're able to get super good results with maximum efficiency, so with minimum cost. And this is really relevant because the competitors, they spend a lot of money training their models, and we're able to get just the same thing with a 60% reduction cost on the training. And this is a lot, because we need to feed more and more data to these guys, because there's a myriad of different sequences that are sitting out there still, and that we want to feed to create better representations for our models. As I said, these models create better representations. Better representations mean better models downstream, so more pockets, better drugs, right? The better the representations, the simpler these models can be. So the simpler they can be, the more efficient they can be, so we can do predictions at the large scale. So 60K pockets to billions of different chemistries in a matter of just days. So as I was saying, our models learn from this protein language model, from these representations, and basically they are able to do 3 different things. One is finding these guys in there. These red guys are new cavities that we're able to find in these proteins where we say, hey, there's a region that may be susceptible to bind drugs that might have been discovered or might have not been discovered yet. The other thing that we are able to do is to create these spaces of drugs, active compounds, so drugs that will be useful, drugs that will not be useful, and to pair them into the same region of this space, of this vectorial space, that basically will tell us, okay, so when you go to the lab, just go to test these molecules because the other ones will not work. And we're able to do that on this scale, so 50 billion pairing in just a single day or now on one GPU. And the other thing that we're able to do, as I mentioned, is to simulate thousands of different structures to create these conformations, these different snapshots of the protein, to, again, search these binding regions into the protein and assess billions of chemistries into that. So now we're sitting on a technology which is very exciting, which we know that it works, but now we want to scale it more. As I said, there's a lot of data that is sitting out there, but we haven't used yet, but we need more compute. So we need two actions here. One, train better representations, because it gives us better predictions on the pocket side and on the drug target interaction side. The second action is ramping up the simulations. The more data that we have on these conformations, the better that we will be on predicting these conformations at a lower cost. So we have a one-year roadmap designed for doing that. We want to first produce this scaling of our technologies to create a gap even bigger than the one that we have now to other competitors. Then to create this human atlas of human drugability in phase 2, and then create this very same human atlas, but with as much conformations as we can for the human proteome. All of these, of course, I mean, I'm the AI guy, but also I like very much to validate everything experimentally, because this is key to retrain our models and to get this gap forward. But all of this, it's accompanied with experimental validation. Of course, this is the beginning for us. We are very aware that drug discovery does not end up only with molecules, but also we will need indications. Now, one of the beauties of doing this technology the way that we do it is that we are not committing with an indication in principle. So now we will focus on just assessing which are the most druggable options, and then basically we will de-risk the decision based on the druggability of our targets and the most interesting indication for us, which will come later, right? So we are just reversing the typical way that early drug discovery is done. So first the most druggable targets, and then the high interest indication. As I was telling you, the market is huge. Since we are not committing now to any compound series, to any indication, to any disease, we can attain virtually, of course, we will not get that market, but we can attain virtually all these different markets, which are really interesting, and they will have lots of return of investment for an investor. So with this, we are now spinning off this department from a spin-off of BSc and IRB to make all of these technologies work together and to scale them up, validating them experimentally, and get this gap to the rest of competitors. We have a technology that is already working. We have super reduced training costs with state-of-the-art performance, and we already have expertise to deliver these projects in real-world programs. So with that, thank you, and I'm open to questions.
Some questions? Yeah, here in the middle.
First of all, thank you very much. I'm excited about your presentation because my job is related to NGS, to testing of cancer. But I'm going to play a bad cop today, and I'm going to challenge you and ask about the cost. At what cost goes all this innovation? Because this is such an irony when we are able to discover a lot of molecules, but at the same time we create a huge footprint which is cancerogenic. Yeah, so how do you deal with that? How do you mitigate this risk?
Actually, that's one of the main things for us. I'm not going to say that we are all about the carbon footprint of our models. I'm just saying that we're all about reducing, enhancing the efficiency of how we learn. As I showed you, our training costs, which are directly associated with the compute time that we use, which is directly associated with the carbon footprint, is super low compared to all other competitors. So we are able to, and we learn super good representations of the biomolecules that we want to work with. So if we are learning at a low cost and we learn super good representations, it means that the models that we build downstream can also be very simple, and simplicity comes with efficiency. So by getting a decent amount of compute at the beginning, which, it comes with a carbon footprint, we believe, and we are actually sure that we can make the whole thing super efficient and then lower the cost and the carbon footprint all in all.
And then, if I may, another question about hallucinations is, well, it's a well-known thing about AI. How do you mitigate this risk of AI hallucinating new molecules?
Yeah, we mitigate that by not using generative models. So none of our models is generative. What does it mean? When I say that we create representations, is that the model is just learning from lots of data and creating vectorial representations that we can later work with. But it does not generate new molecules. It does not generate new proteins. And the molecules that we use, they are made by combinatorial chemistry in the lab. So actually, we have these long databases that we say, okay, so we want to work with these molecules, and then we call our partner in the lab, and they are able to make it experimentally. But we do not create anything from scratch.
So no creativity, like temperature zero.
No creativity. It's human creativity for all the process.
Thank you.